and welcome to the 88th episode in our AI and You podcast series. Today is Wednesday, March 2nd, 2022, and I'm your host, Vicar Saidi. I'm a computer scientist, an engineer, a lecturer, and a consultant. I'm also the author of several books. My most recent book is about artificial intelligence and is titled The Genome Affair. The Genome Affair is a speculative work. It examines what the world might be like if some of the more extraordinary capabilities forecast to be realized in AI over the next 20 to 30 years were actually realized today. Given the growing list of frightening existential threats humankind now faces, the book pays particular attention to the impact AI is expected to have on world affairs. The book is available in ebook format for those who prefer to read on a digital device, but it's also available in a high-quality paperback edition. The Genome Affair is available on Amazon, so I hope you'll take the time to read it and to leave a review. Amazon book reviews are very helpful for writers. I'm very interested in how science and technology influence world affairs and the big questions facing humankind, studying at the confluence of the great disciplines of human history, political science and thought, international affairs, science and technology offers a deep understanding and pedagogically important lessons of how advances in human endeavor have influenced and impacted civilization. I'm available to give talks on artificial intelligence and its related technologies, and on the impact AI is expected to have or is already having on our world. If you'd like to get in touch with me to arrange a web-based event or consulting meeting with your company or organization, you can find my contact info in the podcast notes below. Please make sure to subscribe to my channel, like this video, and share it with others if you find the content valuable. Thank you. In our last podcast, episode 87, I spoke about Vladimir Putin's Russia and the invasion in the Ukraine. In today's podcast, episode 88, I'll speak about AI, genomics, and the potential to accelerate gene therapies. Let's get started. In 2020, Jennifer Doudna of the University of California, Berkeley, and Emmanuelle Charpentier of Max Planck in Berlin were jointly awarded the Nobel Prize in Chemistry for their pioneering work in gene editing. The two scientists and their teams developed what is essentially a molecular scissor, known as CRISPR-Cas9, that can locate and edit genes within the human genome. It was a seminal achievement in human history. The science and technology of gene editing is amongst the most powerful ever developed. It can be used to treat patients with genetic disorders and even to eliminate the disorder using a technique known as germline editing. This will prevent the disease from propagating to the patient's offspring. With a modest level of reflection, we soon realize there are a virtually unlimited number of applications of CRISPR-Cas9 that can be both beneficial for humankind as well as nefarious. The genome is the complete genetic blueprint of an organism's cells, organs, and tissues. The Cas9 protein is an adaptive capability within the natural immune systems of bacteria that they employ to fight any viral infections they might encounter. RNA molecules provide the address of the target, thus guiding Cas9 to the viral DNA, which can then be edited. Drs. Doudna and Charpentier's research has now managed to harness this natural CRISPR-Cas9 molecular scissor mechanism for multi-purpose gene and germline editing within the human genome. In 1990, scientists around the world initiated the Human Genome Project. For the first time in history, they'd started what was expected to be a 15-year-long international research effort to map the complete DNA sequence of a single anonymous human, their genetic blueprint or genome. By 2003, an accurate and complete human genome sequence was completed 2 years ahead of plan. It was an extraordinary achievement considering that the human genome contains 3 billion chemical base pairs of molecules, adenine, cytosine, guanine, and thymine, that would require about 20 phone book-sized volumes to fully document. Of these 3 billion base pairs, 2% yield about 20,000 genes. These 20,000 genotypes manifest as phenotypes or traits in humans. The remaining 98% of the base pairs, what scientists call the non-coding region of the genome, are yet to be fully understood. We are learning that many of the base pairs perform a critical regulatory function. They determine whether and when gene expression takes place. But the question of which of these base pairs are involved in gene expression, and in what combination, or whether some base pairs perform a yet unknown function within the genome, remains a mystery. They may well control how a disease expresses and progresses at the cellular level. Fortunately, scientists have begun to leverage advanced and emerging capabilities in artificial intelligence to help decipher the human genome. As an increasingly superior proxy for human intelligence, AI can be leveraged to solve what may otherwise be insurmountably complex scientific problems for humans. Due to AI's speed and ability to evaluate and extract insights, which from the human perspective would be, for all intents and purposes, an infinite volume of data, long-standing problems in science are increasingly being solved. Today, a modality of machine learning for artificial intelligence, known as a transformer, can evaluate ever larger sets of ACTG, adenine, cytosine, guanine, and thymine sequences in the genes and non-coding DNA within an individual's genome, and then predict how these genes will manifest themselves in cells, organs, and tissues. This data-driven predictive capability offers a dramatic advance in human understanding of disease and in developing the ability to contain disease, enabling gene expression. Cystic fibrosis, Huntington's disease, muscular dystrophy, and sickle cell disease are all well known for having a genetic basis, and we may soon acquire the ability to manipulate genes to prevent or reverse these conditions. We know that some humans do not develop high levels of cholesterol, even though they may consume a diet that normally results in an elevated cholesterol level in most others. Thus, we may be able to edit the regulatory gene using CRISPR-Cas9 so that we can control cholesterol production rather than be subjected to traditional chronic pharmaceutical therapies involving statins. This approach may also be more cost-effective, have fewer side effects, as well as provide superior clinical outcomes. In summary, artificial intelligence can lead to a significantly better understanding of biology, and this can lead to a dramatic improvement in healthcare and medicine. We are evolving from simply trying to observe biology to leveraging it to prevent and manage disease. AI is increasingly enabling us to wrap our heads around what is otherwise an unmanageable level of complexity in genomics, while advances in genetic research simultaneously improve our ability to predict how DNA sequences influence gene expression. Thank you for spending some time with me. I'm trying to follow the TED Talk format, and so I'm keeping these podcasts under 20 minutes. Let me know what you think. I hope you'll find these insights into artificial intelligence helpful, and I hope you'll read my new book, The Genome Affair. It's on Amazon. Until next time then, this has been the AI and You podcast with author Vicar Saidi.